Alright, so we actually did this in back then in our biochemistry days, but we are back here talking about this in microbiology. So look in depth at the structure of an immunoglobulin. Alright, immunoglobulin. So we're just like trying to maneuver from the picture, go to the shape, the parts and all of that. Right. So immunoglobulins are also known as antibodies. So they are specialized glycoproteins produced by plasma cells because you want to know what the body wants to recognize and neutralize the bad guys. Those are the pathogens. So their structure is actually what's crucial for the function they perform. So this is the structure. Now looking at this whole structure, it looks like a Y. Okay, it looks like a Y. So that it's Y-shaped molecule. So it's composed of two heavy chains and two light chains. So they are held together by disulfide bonds. So see all these S to S, 2S, 2 sulfide, disulfide. You understand? So they have a variable and constant region. That's the V and what the C. So you see the CH1, Cl, VH, VL, all of that. So the V region determine antigen specificity, while the C regions determine what define antibody class and function. They also have the Fab area. Now the full meaning of Fab is what? Fragment antigen binding. Right, so where's the Fab area here? Look. Alright, where's the Fab area? It's not labeled here. Well, this is the antigen binding site though, but let's look where they actually label the Fab. So don't really, okay, yes, Fab region, Fab area. Do you understand? So it contains the antigen binding site. Then we have the FC site. It is responsible for what immune system interactions such as what complement activation and binding to immune cells. So these are the different types of what immunoglobulins, which we have already spoken extensive on it. I told you guys that the mnemonics to remember this is GAMD, right? So you can see all their functions there. That's basic too much, right? So if you want to look at it, you can just pause the video to look at it, right? So that's it for the different structures or parts of immunoglobulin.